video is going to be on sections .2 to .4 of chapter 22, what has to do with speciation. So, and a large part of what we looked at so far is what we call microevolution. So that was the change in allele frequencies in a population over time. So in microevolution, new species aren't being formed. Rather, the variation within a species is increasing. Traits that are beneficial to that species' survival are becoming more prevalent in the population in terms of allele frequencies over time, but the species is remaining the same. So within the medium groundfinch population in the Grant's experiment, even though beak depth was increasing over time in response to the drought through natural selection, those medium groundfinches with the deeper beaks were still medium groundfinches. They weren't a different species yet. So that's what we talk about in microevolution. Speciation is an example of macroevolution, or how microevolutionary processes can accumulate over time to create whole new species. So to really talk about speciation, we really have to understand or have a common definition for what a species is. So for our purposes, we're using something that's called the biological concept of what a species is. So this is kind of called the biological concept. And the biological concept defines a species as a group of populations. So a group of populations, all the same species, that are capable of interbreeding and producing viable, fertile offspring. So these groups, these populations of the same species have to be capable of mating with each other, and that the product of their mating is able to develop in the womb, be born into a living, viable offspring that then is fertile and can reproduce. So, if that is our definition that we're going to use, then speciation becomes the evolutionary event where the small microevolutionary changes build up over time and cause new distinct species to develop. So, one species, ancestral species, to diverge into two new species. So this is speciation. For speciation to occur, there must be what's called a reproductive isolation. And reproductive isolation is where members of one population of a species are prevented from mating for some reason with another population of the same species. So we have population 1, population 2, and something prevents them from mating. So there's no mating between members of these two populations. That's reproductive isolation. Your book talks about a few other definitions of species. These are interesting to look at and to see the differences between biological concept and these concepts, but it won't really be necessary for the AP exam at all. So there's several things or factors that underlie speciation, which by that I mean these factors are things that cause reproductive isolation. Generally, we categorize all these different factors that can cause reproductive isolation as prezygotic or postzygotic, depending on when they occur. So prezygotic isolation is any force, anything that prevents two populations from actually mating with each other. So these individuals can't even be in the same place, and they can't even mate and fertilize a viable egg. So prezygotic barriers examples are habitat isolation, two populations live in completely separate habitats, located spatially a distance apart, so it's hard for them to be together. Temporal isolation, so maybe one of the populations or species is only out at night and one's out at the day. Behavioral isolation, something about their behavior separates them. Mechanical isolation, there are physical barriers like mountain ranges, shells for these snails. And then lastly, gametic isolation. So all of these are prezygotic barriers that prevent two organisms of a given species for even coming into contact to mate. And the second type is postzygotic isolation. So once, two individuals of a species actually can mate with each other, but something happens after fertilization that prevents development, birth, growth, or fertility of the zygote. So one of the things is if they do reproduce and give birth, the fertility of the offspring, they're sterile. Most hybrids, which we'll talk about in a bit, are sterile. That's why they're like ligers aren't a different species. Another thing, something might happen, or things usually go wrong in development of the fetus, of the embryo, and that's why fertile offspring can't be formed. And third, if a hybrid is born, there might be so many different physiological things that aren't working correctly about it that just breaks down before it even reaches the age that it could produce offspring itself. So prezygotic, postzygotic are different factors that cause reproductive isolation. So, if we have reproductive isolation, how can we actually form a new species? And there's two main types of speciation called sympatric and allopatric speciation. And they differ by how reproductive isolation is achieved. In this first type, an allopatric speciation, a physical barrier, such as an ocean or a mountain range, prevents reproduction from occurring between two populations. Why does that need to occur? Well, so that any traits or novel genes that develop in one population don't flow in between, so no gene flow is happening. So physical barriers are present. So, for example, if we have an ancestral rabbit species that live in this area, and over time, this river forms and widens and geographically separates two populations of that species. Any mutation that arises in either species from natural selection won't flow into the other species because they can't really cross this river to mate with each other. As time passes and the two species diverge via natural selection enough, some of the genes that are affected by this natural selection may involve mating, and then even if the two species were put in the same area, they wouldn't or couldn't reproduce with each other, and two new species have occurred. So we know speciation has occurred once you can take the two separated populations, put them back together, and they won't breed or produce fertile offspring. So some examples of allopatric speciation is the prevalence, the radiation of all of these different types of lobster down here in the Caribbean and the North Pacific Ocean. They came from one ancestral species that migrated before this Panama land bridge was here. And after the Panama land bridge was formed, the different populations of that species were cut off from interbreeding with each other, and via natural selection developed their own novel traits and speciated, as you can see here. The second type of speciation is called sympatric, and sympatric occurs when two populations of a species become reproductively isolated and diverge without a physical barrier, so no physical barrier. For example, these fish, allopatric speciation, here's this physical barrier. Sympatric can still migrate over the barrier, but we still diverge. How is that possible? Well, there's some different ways. One is something that's called polyploidy. So remember, we talked about that in non-disjunctive events, we can have two extra or few chromosomes. Well, in the past, organisms have evolved pretty much instantaneously by having a mutation which caused their chromosome number to double. Meiotic events didn't happen, so we didn't have segregation of homologous chromosomes, and an organism ended up with tetraploid 4n instead of 2n, and that led to immediately reproductive isolation because those tetraploid organisms couldn't mate with their other diploid members, and so new species evolved. This is rather rare in animals, but in plants it's super common. Almost 80% of modern plants have evolved by polyploidy. Sexual selection is a possibility. So, let's see. Cichlid fish in Lake Victoria in Africa have evolved sympatrically by sexual selection. Certain females started only selecting lighter colored males, and over time, this behavioral pre-zygotic isolatory mechanism via sexual selection caused the divergence of species. And habitat differentiation is also a big mechanism. So habitat differentiation is when populations of the same species diverge because they utilize different resources in the same location or develop adaptations to different niches in the same location. So for example, the green tree frog in the United States diverged sympatrically because different populations started to adapt to conditions in different niches or different locations in their zone, in their habitat here in central eastern the United States, and so they eventually diverged. A special kind of habitat differentiation is called adaptive radiation, and this is when one parental species quickly gives rise to many new species when different populations of that parental species end up developing different adaptations to specific niches of their environment, and so they diverge. And this is what happened with the finches in the Galapagos. One parental species ended up diverging or radiating to all of these new species because each species began to adapt to a specific niche based on food source of the same environment. They at first weren't necessarily geographically isolated, although later there was some allopatric speciation, but initially they just became adapted to different food sources, and so were spatially separated by where that food was most plentiful. So sometimes two distinct species do mate with each other, and they create what are called hybrids, like a lion and tiger create a liger. Ligers aren't fertile, they're sterile, that's why a liger is not its own species, but the resulting hybrids usually occur in zones. They're called hybrid zones that are like in between buffer zones between the environments of either species. So for example, in Central Europe, we've got the fire-bellied toad and yellow-bellied toad. One lives in Eastern Europe, one lives in Western Europe, and this border in between is the hybrid zone, where hybrids between these two species mating occurs. Since hybrids are either sterile or have significant physiological problems, they rarely reach maturity and don't really reproduce or mate with other species, cutting off that gene flow. However, sometimes hybrid zones can go through some different kinematics. So, if these hybrids are perfectly fertile, we have gene flow in between the species, and that species ends up fusing. So if enough of those hybrids form and go back to their respective populations and mate with those, eventually over time we'll have a fusion of species into one, and we'll kind of get rid of our two species. Hybrid zones can also reinforce a gene flow barrier if these hybrids are significantly less fit than either of those species. Then that'll increase the reproductive isolation of those two species and cause them to diverge even farther. or if the hybrids, continue to happen, are less fit, so they don't limit gene flow, but they're not extremely lethal. They might just stabilize or continue the genetic differences between the two species remains kind of stable. Hybrids keep happening, but they don't contribute a lot to gene flow. The last thing we want to talk about is the rate of speciation. So how long does it take for new species to form, and why does it seem like some species just appear in the fossil record? So macroevolution exhibits a gradualist trend, meaning species gradually evolve over time via the accumulation of microevolutionary changes. So over time, the microevolutionary changes build up and build up and build up between separated populations, and eventually those changes become significant enough that they are bona fide new species. However, gradualism is also often punctuated with periods of rapid evolutionary change. So sometimes it might not seem for millions of years that a species is really changing that much in the fossil record, and then boom, all of a sudden there are periods of rapid change where a number of speciation events happen. That's called punctuated gradualism, where for periods of time we have gradual evolutionary change punctuated with intense periods of change. And this is really caused by, if you think, what does natural selection do? It forces individuals to adapt to their environment, promotes best adapted to survive. Well, if there's things like natural disasters or significant environmental changes, that might be followed by a period of rapid evolutionary growth, or rapid speciation, as adaptations are much more beneficial if it's a situation that's very dire, where there's low food or sunlight or shelter. All right? And one example of this is, again, those cichlid species. What's happened now is, via pollution, the waters are becoming muddy in Lake Victoria, or muddied or murkier, and those females can't distinguish the coloration patterns of the male fish, and so can't exert preferences. Instead of having a clear color difference, they kind of look monochromatic in this orangier polluted light, and so that species is actually fusing back into one species because that sexual selection isn't possible, that prezygotic isolatory barrier isn't possible. And that's happening pretty rapidly. So just over your lifetime, you might see that species in Lake Victoria fuse. And that's an example of rapid speciation due to a drastic environmental change. All right, that's it. We'll look at this in class. If you have any questions, let me know. But otherwise, have a great day.